Hello everyone, I am Shurasri Pal and we are discussing this question from GATE Examination 2010, paper Biochemistry, question 12. And the question states, match the items in group 1 with those in group 2. In group 1, the items are P, isotype switching, Q, clonal energy, R, class 2 MHC, and S, self-tolerance. In group 2, the items are 1, 3H domain, 2, non-responsive to self-antigen, 3, non-responsive TH cells, and 4, beta 2 microglobulin. For more information, you can follow this NPTEL course, Immunology. Now, we will discuss these items one by one. First of all, we will talk about isotype switching. So what is isotype switching? Isotype switching is the mechanism by which the antibodies they can change their class, which means that the antibodies change their constant region. Now, the antibodies we know that they have a variable region and a constant region. The variable region is for binding to the antigen, whereas the constant region is for its effector function. So the FC region or the fragment crystallizable or also known as the constant region of the antibody, it is for the effector functions of the antibody. Like what are the effector functions? Like it can be opsonization, it can be neutralization, or it can be um, like opsonization, neutralization, and also complement activation. These are the effector functions of an antibody. Now, the constant region mediates this effector function, and the antibodies they have different constant regions. So, we know that there are different types of antibodies or immunoglobulins like immunoglobulin A, immunoglobulin E, immunoglobulin G, right? So there are these different types of M and D, right? So these are the different types of immunoglobulins, and these different types of immunoglobulins are actually uh, they are differentiated on the basis of their constant region. So they have different constant regions, and these are the classes of the antibodies or the classes of the immunoglobulins. And when these antibodies they switch their classes, that is called the isotype switching, or it is also called class switch recombination. It is called class switch recombination. Now the question is, how does the antibody switches its class? So there is this recombination process that occurs. So we know that an antibody gene can have different types of the constant regions. So this C actually denotes the different types of constant regions. Like here you can see it's C mu, it's C delta. These are the C gamma regions, right? This is C epsilon. This is C alpha. So actually, mu, C mu stands for IgM, C delta stands for IgD, C gamma, these all stand for the IgG, and C epsilon stands for IgE, C alpha stands for IgA, right? So these are the different constant regions, and in these constant regions, so these are all in a stretch of DNA in a single gene. Like these genes are all together on a single piece of DNA, and what happens is that recombination occurs. And so there are actually these switch regions. So these small ones here you can see this blue one, this yellow one, these are the small switch regions where the recombination takes place, and the DNA in between them is looped out and it is excised out. Okay, so here you can see that this C gamma 3 is actually retained in this antibody. So uh, therefore, because of this switch region recombination, you can actually produce Ig gamma 3 or IgG3, right? Now, Here we can see that the C mu, C delta, S gamma 3, and S gamma 3 is the switch region, C gamma 3, these all are actually spliced out or excised out. So the next one is C alpha. So therefore, this antibody will be actually of IgA isotype. Okay, so here we can see the production of IgG3. Here we can see the production of IgA. So this is how the isotype switching occurs, right? Because of this particular process of class switch recombination, and this process generates the different classes of the antibodies. So isotype switching is in the item P, but the problem is there is actually something wrong in this question because isotype switching we cannot match with any of the options because what I assume here is that it should be this VH domain should actually be the constant region or the constant domain of the antibody. Then we could have matched P with 1. Okay, but there is a mistake in the question. There should be a printing mistake in this question. Okay, uh, but don't worry. We will move on to our next topic, which is clonal energy and self-tolerance. So, what is central tolerance? So, central tolerance is limiting the development of autoreactive T and B cells. We know when our immune cells, when they develop, when they mature, there is a lot of process that goes on to rectify those T and B cells. What do I mean by rectify? So actually, they are trained in such a way that they should not react to the self antigens. So they know all our body's antigens. So they are trained and they are trained in such a way that these T cells should not select or should not react to our body's antigens and